Let us learn about the brain. Hi, the topic of this lecture is locomotion and CPGs, which, as we will learn in a second, is an abbreviation for central pattern generators. And this lecture can also be thought as small networks part 2. It's just about particular type of small networks. So basically we're still thinking about how with a few neurons we can achieve great results and build some useful networks. But the question that we'll be asking is, how do animals move? And let's start a little bit from afar by talking about a knee-jerk reflex. The official name of it is a patellar reflex, but you have probably also heard the expression a knee-jerk response. So basically that's what we're talking about. And maybe somebody had performed it on you when you visited doctor's office. Unfortunately, it's a virtual lecture, so I cannot give you one of those hammers and ask you to make an experiment and actually demonstrate a knee-jerk reflex in you or your partner. That's very interesting and relatively easy to do. So if you want to just look it up, if you want to try it at home, I believe you probably can do it with a ruler or something. But at least let's talk about it theoretically. So it turns out that in our knee, there is a special part called the patella, this one. This is the knee itself, one bone, the one that goes to your body, and another bone, the one that goes to your foot, and there is a joint in the middle, but this joint is protected with a special bone called the patella, kneecap, and this bone is hanging on two ligaments, on tendons, that are connected to some muscles and some bones. It's actually connected to the bone here, but it's connected to a muscle over there. And just because there is a tendon that's almost exposed and a bunch of fat over here, some squishy substance under it, if you hit, a better word tap, hit is too violent. You don't want to hit it. You want to like tap it with a special hammer. If you gently tap it, then the leg or the foot, let me try to show it, the foot will kind of go in this direction. Can you see the artist's rendition of the foot? I'm not sure, but the leg goes in that direction, as you can see on the video. So the question is, why do we have it? Why do we have it? Let's think about it. As we tap the tendon below the patella, which muscle gets pulled? Because what happens when you tap on the tendon, it's almost like jerking this tendon, like taking it and pulling it real fast, because when you tap on it, it kind of bends in this direction and it becomes a little bit shorter. So it pulls simultaneously on this part and on this part. Which muscle gets pulled? Let's look at this human sitting on the picture. It looks like, if you think of where the tendon goes, it goes somewhere here. It's obviously connected to the bone up here, but which muscle may it be? It's probably one of the muscles in the thigh, right? It's probably one of the muscles that lift this leg up. That's one of the muscles that you use when you walk. Next question, what is the response? So what happens here when you briefly pull on this muscle with a tendon using a hammer? How does the muscle respond? Does it contract or does it relax? What do you think? It contracts, obviously, right? If it were relaxed, basically nothing would have happened, but it contracts, and that's why the leg moves in this picture towards us. The leg kind of does the knee jerk. So now we have this story. You pull on the muscle and it contracts. You try to elongate the muscle a little bit and it contracts in response. Now, the most important question perhaps, why? Why do we have that? Obviously, it hadn't developed evolutionary so that doctors could test it. Doctors test it just because it's one of the few tendons that you can actually tap on. Maybe there is something in our elbow, I'm not sure, but there is definitely something in our knee. So they use it because they have an opportunity to test our reflexes. But why do we have this reflex to begin with? What's the point? And let me restate it. You have a muscle, you pull on it really quickly and not by much, and it contracts in response. Why in real practical life it could be useful? Pause the video, think about that, then compare your answer to mine. Basically, if you think about it, you pull, it contracts. It counteracts the pull, right? It feels like a fast negative feedback thing. So you have a muscle, you pull on it, and it contracts back. It's as if muscle didn't want to change whatever length it used to have. And that's exactly what's happening. Why is it useful? Because if you restate what's happening in slightly different words, it will almost provide us the answer. So when the muscle is of a certain length, if we elongate it, it again becomes of the same length because it contracts. Which means that if the foot was in some shape, can you see it? I should draw in different part. If the foot was in a certain position and now you pulled on this muscle above, maybe because the foot changed its position, what will happen is that this muscle will get activated and it will move the foot again, returning it closer to its original position. Which is useful, right? The leg is trying to preserve whatever posture that it had naturally without you trying to do that voluntarily. Because the funny thing about this reflex, and again, I'm so sorry we cannot just run it virtually. The funny thing is that you don't feel like you move your leg. The funny thing is that it feels like the leg moved itself. Your body, without you consciously trying to maintain a posture, tends to maintain the posture just because these reflexes exist. And again, they actually exist in every muscle. This is just the only muscle that's easy to manipulate in a doctor's office. We'll come back to this part of the story of how it is useful in a few minutes. Let's first talk about the third question. I should probably sit a little bit lower so that you would see it above my head. What would be the minimal neural circuit to support it? How many neurons do you need in order to create a circuit that would support this kind of response? Pause the video, think about it, maybe draw the minimal circuit. Is it 1 neuron? Is it 2? Is it 5? How many do you need? What neurons would it be? And how would they be connected? Let's assume that we know where it all ends in a muscle that contracts. And let's assume that we also know where it all starts. I will tell you where it starts. There is a special sensitive fiber in the muscle that senses the length of the muscle and reports it to the nervous system. Use this information in order to sketch the minimalistic circuit that would support this reflex. Alright, here's how things happen actually. We have a special cell in the muscle, a sensory organ that senses the amount of stretch in the muscle. So there is a receptor in the muscle, it talks to a neuron, and neuron sends its signals to the spinal cord. This part is a cross-section of the spinal cord. You wouldn't know if you've never seen it before, but if you take a human, this is a human, and the human has a spinal cord inside, and if you make a cross-section of the spinal cord, then because it's essentially a tube, it has a certain shape to it. So this is this shape. We have a sensory neuron that talks to the spinal cord and two motor neurons. One motor neuron talking back to this muscle and another motor neuron talking to the muscle on the other side of the leg. These muscles are complementary. They move the leg in two different directions. One is extensor, it extends the leg, another is flexor, it flexes it. For the muscle itself, it's pretty much a feedback excitation. Let's see what's happening here. Muscle is elongated, and this thing that's sitting in the muscle, the stretch receptor, detects this elongation, activates the neuron, and now neuron spikes, and activation is running along this axon into the spinal cord. In the spinal cord, it synapses on a motor neuron that returns this activation back to the muscle. So what happens is a feedback loop, and it is sort of a negative feedback, even though it's a case of feed-forward excitation. Muscle is contracted in response to elongation. I guess what makes this feedback negative is that the receptor detects elongation and causes contraction. That introduces this minus sign, makes it a negative feedback. Elongation leads to contraction. But at the same time, another interesting thing happens for the complementary muscle on the lower side of the thigh, the flexor. The same neuron that sends information to the spinal cord also activates an inhibitory interneuron, and this inhibitory interneuron inhibits the motor neuron that goes to the flexor. It doesn't necessarily shut it down completely, but it makes its spike less, and this also helps to move the leg back. In this case, it's not quite moving back, it's making it like move in a funny fashion, but in real life, as we have just discussed, when this reflex helps you to maintain posture, the fact that the reciprocal muscle gets a little bit relaxed helps you to maintain the posture. if some external force tries to bend your leg a little more, the leg kind of automatically bends a little less. Now, equipped with this knowledge, we can face a bigger question. How do we move? And let's look at the example of running. How do we run? So what I want you to do here is to introspect. Imagine that you're running. Imagine that you're running cross-country on some uneven surface on a little dirt road. Imagine yourself running and try to just think about what's happening in your body. The first question is, do you control every step consciously? Do you? If you're running a 5 miles run, you're running for a while now, for the last 15 minutes or so. Second, what happens if suddenly you step on a little stone and the stone rolls? Like you don't completely trip necessarily, but you step on something and unexpectedly moves, or maybe you step on a route that you haven't noticed. What happens with you as you run? If you ever experience that, you can kind of almost remember how it feels and try to introspect what may be the mechanisms behind that. Third question, what do you do? What happens to your running if suddenly you have a little stretch that is slippery? So maybe you're running late in the fall and it's below the freezing point already. And so now you have this segment where you have black ice. What changes about your running? And finally, what neural mechanisms may underlie each of these aspects of running that you can introspect? Pause the video, think about it, maybe make some sketches on a piece of paper, and then we'll talk about that. Before we go for the answers, let's look at 3 pieces of additional evidence. Don't forget your answers if you haven't written them down, like remember them, we'll come back to that. But let's look at 3 pieces of evidence that we have. One piece of additional evidence is that headless chicken can run. That sounds weird, but that's apparently what actually happens. If you chop chicken's head off, and people do that when they prepare chicken soup, even though it sounds gruesome, it's part of life for those who eat chicken. When you chop chicken's head off, the body can still run for a while. Obviously, it falls down eventually, but they can even fly for a few minutes. That's one piece of evidence. The second piece of evidence is this really old video. Apparently, if you take a cat, and that sounds even worse than a chicken, and I'm sorry. It's an old experiment that I totally don't endorse. But back in the day, the ethical sentiments were different, and experiments that wouldn't be popular now were quite mainstream. So back in the 60s, people discovered that if you take a cat and then you make an incision in the nervous system, so basically you separate the brain from the spinal cord, this cat is practically paralyzed because the brain cannot talk to the spinal cord, which means that the cat cannot move any of its muscles voluntarily. However, the spinal cord is still there. And if you suspend this cat, because it cannot stand on its own, but if you suspend it and then put it on a treadmill, the legs, the paws move, making the cat kind of walk on this treadmill. The paws of a cat move in this walking fashion and even adjust to the speed of the treadmill without any influence from the brain on the spinal cord alone. It's enough for the spinal cord to be there for this walking motion. Here's another piece of evidence, much less gruesome. Babies can walk a little bit. I mean, they cannot really walk, but they have reflective walking in newborns that are only a few days old. I mean, if you've never seen a newborn, it may sound like amazing. It's actually not particularly amazing. It's pretty pathetic if you think of it, but when one of their feet is stimulated, they move the other foot forward. As I was looking for a good GIF, I also found this GIF, which I'm pretty sure is played at a much higher speed compared to how it was recorded. But yeah, there is a little bit of this walking. It's a reflex that pediatricians actually check to check whether the newborn is developing properly. Another piece of evidence, gaits. A gait is kind of a style of locomotion. How many gaits do humans have? Like you can walk fast. Are these different gaits? No, this is the same gait. You just perform it at a different speed. Walking is a gait. What other gaits do humans have? Running. What's the difference between walking and running? In walking, in a human, you always have one foot on the ground. So you move one, then another. When you run, there is a moment when neither of your feet are touching the ground. You are suspended in the air for a very brief period of time, but this suspended period repeats again and again and again as you run. Do humans have any other gaits? Basically not, at least not naturally. There is one artificial gait that you can make that's not used by normal humans. Skipping. So skipping is kind of like an artificial gait. In the same way as horses have natural gaits, but then there are trainable gaits that you can train a horse to do for the sake of competition in like how well trained this horse is. But what are the natural gaits in a four-legged animal like a horse? Is it also only two, walking and running? It turns out that in a horse with four legs you have more options. This is a diagram that I found online that I don't completely understand. I just really like how it looks like because it's so complicated and fancy. Supposedly it's a diagram of different gaits. The basic gaits are walk, and walk in a horse is kind of similar to walk in a human in the sense that you always move one foot at a time, one hoof at a time in this case. And gallop is analogous to human running because there is a phase of full suspension. There is a phase which is easier to see in the previous video actually, in the case of a dog, or this dog. There is a phase when the animal is fully suspended in the air. But then there are also two other stages in between because again, four legs give you more opportunities for changing the way how you move. There is pace and trot. Trot is actually what most horses do, and in trot you move your front right leg and your back left leg at the same time. It's kind of hard, it requires lots of coordination to show it with fingers, but that's kind of like, if this is a horse, it moves these two legs together. Oh god, it's hard. It moves these two legs together, and then it moves these two legs together. And it's called trot. Some horses don't trot, they pace, which means that they move both legs on one side together, and then both legs on the other side together, and move like that, kind of. And then there is also canter, that's in between trot and gallop. It's interesting because that's when neuroscience kind of bleeds into medieval history and fantasy. Because, for example, pace, that goes like that, instead of doing trot, apparently pacers were valuable back then because they are easier to use for carriages. Like that's how specialized horse breeding can be when you actually use horses not as ornamental animals but for practical reasons. You actually select horses that tend to pace because they can be used in some situations where animals that trot don't. And if you look at six-legged animals, they have even more freedom, and that's not something that most people usually think about, I'm guessing, but a really slow beetle walks, and then a faster moving beetle runs, and then a really fast beetle. Not all beetles can gallop, but some of them can, and people study that. I don't completely understand these pictures, but they clearly show beetles, and they clearly show these diagrams on when each of the legs touches the ground. So it must be cool to be a specialist in beetle gaits, isn't it? Beetle galloping. Now we can integrate all these evidences, all these pieces of evidence into one story. Let's say we have a leg of some sort. Let's draw like human-like leg, but it could as well be a horse-like leg. And we have a spinal cord. Okay, so somewhere here sits a spinal cord. And then we also have a brain, so that's also good. I'm out of space now, so this would be a brain. Sorry, brain. You're a little bit to one side, I'm afraid. So, what are the parts of this story? First of all, there is the knee-jerk reflex and other reflexes of this kind. So when you have a muscle, there is a system of reflexes that makes sure that whatever commands were given to the muscle, it stays roughly at the position, or rather it is activated roughly to the extent to which other systems want it to be activated. That its length. That's the best way to say that. Its length is as it's supposed to be. If some other force tries to artificially elongate the muscle, it gets activated a little bit more. And the other way around. If some force tries to shorten it, it gets activated less, and so it's allowed to elongate. How is it useful for our running introspective exercise? That's what happens actually when you step on a little stone and the stone suddenly moves from under your leg, or if you step on a little root that you haven't noticed. You expected that you will hit the ground here, but you actually hit it at a slightly different height, which means that your muscles are not at the position at which your nervous system expected them to be, and you have an immediate correction. It doesn't lead to these cascading consequences. Nothing breaks down. I mean, at some point it can, if you step on something really bad, really large, it may throw you so off pace that you will fall down. But for most disagreements between what you expect and how you actually placed your foot and your leg, you can probably correct for that. And you don't correct consciously. Let's go back to this first question. Do you consciously plan every step? No. Right? If you're running for 15 minutes, you're just, okay, I'm running here, I'm running there. So you're in this mode of running. Can you control your feet if you have to? Yes, you can. There have to be some way of controlling your feet. So there have to be some part in the brain that sends signals directly to the foot, except actually we don't have signals that go directly to the foot, but that's just how we vertebrates, that's how we are. We have motor neurons in the spinal cord that send signals to the muscle. And these neurons get inputs from the muscle itself, but they also get inputs from the brain. And in principle, brain can override whatever is going on and do a voluntary motion of the leg. But so then how do you run? When you don't think about it, when you don't move your legs consciously, what happens? You have this rhythm going on. And from the Gait story, we can realize that these rhythms are slightly different. We can run faster, we can run slower, we can switch to a walk, or we can go running again. In case of horses, because they have 4 legs, and I guess it's harder to control 4 legs, they have less control over where the legs go, or at least that's my impression after reading books. I've never actually seen a horse. Well, I've seen it from afar, but not close because they have teeth and I don't like that. But horses apparently can damage themselves pretty badly if, for example, they go on an uneven field, on a field where groundhogs had made some holes, because a horse cannot process information fast enough to change where its foot goes. If its hoof goes into the hole, it will just break its leg, and that's very sad. So the horse it almost feels like driving a car, if you think about that. Just the very way the horse moves as it switches from one gear to another gear. So here it's pacing, or like here it's trotting, and now it's in canter. And now you're like, let's go gallop, and you go gallop, and you can do gallop for only 2 minutes. I don't know how long. Horses cannot gallop for long. It's an emergency mode. But either way, in order to maintain this rhythm, there have to be something that generates this rhythm. So there have to be a rhythm generator. And let me draw it as some metaphorical cycle, just to show that something is going on in a circular fashion. It's happening, and then it's happening, and then it's happening, and then it's happening. Pum, 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 pum. If it's made of neurons, and it is made of neurons, then you can take one of those neurons and also connect it to this neuron. If this thing is running, then this muscle, this motor neuron, and because of that, this muscle will get activated again and again. And so if you have another muscle, you just make sure that it is activated at a different phase, right? Because when you activate the muscle that extends the leg, it's not the same moment of time when you activate the muscle that flexes the leg. You probably first extend, then flex, and then again extend, and then again flex. So there will be some other neurons, some different place that will activate some other neuron. I'm out of space in my spinal cord, so I'll draw here. That will activate this other muscle. Can you change the speed? Yep. Can you do it voluntarily? You actually probably mostly do it voluntarily. So somewhere here in the brain will be another signal that will go and it will control this cycle, this loop that goes again and again and again, that loops through the pattern of activation and activates all muscles in the correct sequence, because obviously the leg doesn't have only 2 muscles, right? There are more muscles here and all the toes. There are many muscles in the leg. I'm not sure how many, but you can check in an anatomy book. This happens, and also either this loop that rotates can be switched, not only its speed can be slightly adjusted, but also it can be switched into different profiles, so that we could run sometimes and walk sometimes, or maybe there are more than one. Maybe there is one for running and one for walking, and we can also switch between the two, so we can engage one or disengage another, or maybe they're partially overlapping. That's roughly what's happening. That has to be happening in order to support the motions that we have. We have reflexes, we have control over the motion, we have this thing that generates the rhythm, and we have control over this rhythm generator. We change the way it operates kind of the same way as you drive a stick shift when you move from one gear to another. Is it unique for humans or for horses? No. In fish you have the same thing, except in fish there is only one gait, probably. Swimming, because fish have the record low number of legs, 0, but they also can swim fast or they can swim slow. And they also have all these reflexes. Remember, we just talked about escape reflexes. They may be swimming, but then something else maybe happen, like they may turn a little bit while continuing swimming, and then the turn will organically transition into a new type of swimming and so on. And for this swimming, you also have kind of like a gear shift. It's just a little bit more continuous because you can swim faster or you can swim slower. And that's nice. It's nice to know that you can study that in fish. It means that you don't have to dissect horses. It would have been horrible. I would have been really against that. That was a joke, but it's also not a joke, because that's how ethical research works. We always try to go for a minimal possible preparation, right? We try to reduce harm. If we need to study that, and we need to study that probably, even if you're not interested in neuroscience for the sake of neuroscience, there are reasons to learn how people walk, because there are all sorts of disorders and all sorts of trauma when people have trouble walking. All of these discussions about how our spinal cord helps us to walk is really important for people who have some issues with the spinal cord. And by the way, one of the things that I kind of forgot to mention, how does the decerebrated cat and the headless chicken play into that? These observations show that the part that generates the rhythm has to be in the spinal cord. And indeed it is there. So when you run, the rhythmic motion of your legs is actually generated not by your brain, but by your spinal cord. And in a fish, you can study that, and you can even record from an isolated spinal cord. You just take this little piece of the spinal cord and you connect to it with electrodes, and you see that there are neurons that are active and then not. These are bouts of swimming, but within each of those there is periodic activation of muscle that would create this undulating motion. So you don't even need a full fish to study that. It's enough to have these teeny tiny zebrafish larvae, which have just hatched and are a millimeter in length, and you can work with the spinal cords. You can just cut this piece out and study how it works. But do we need this complex system with all these reflexes and whatnot? Cannot we just control everything from the brain? It turns out that it's pretty smart to delegate some of the work to these lower hierarchies, so that the brain can deal with general ideas of where to walk and how quickly to do that, and only on special occasions it would override the control of a leg, so that you would step precisely somewhere. As people in early 2000s developed robots, it was actually quite noticeable, because some groups of researchers tried to make robots where each leg is precisely controlled by a central system. And one of those robots was Honda's Azima robot, and there is this famous video on YouTube where it's very sadly falls from the stairs, because climbing stairs, as it turns out, is really hard for a robot. And that's mostly happened because all of these motions of this robot were coded, and they had to be kind of perfect. And if there was a disagreement between the reality and what the robot was trying to do, the disagreement would only grow over time. So there was not enough negative feedback. If the robot started falling down, it would continue falling down. But then Boston Dynamics, and you might have probably seen these videos just because they look so ridiculous, the whole point of this group was that they tried to create robots that can be pushed around, that can step on ice, systems that first try to recover and only then try to control where they move. So they kind of have reflexes coded in, of course, similar to our knee-jerk reflexes. As you can see, when this person pushes this robot, and there are lots of funny videos on YouTube, when the robot loses its ground, it tries to come to the posture that it's supposed to be. And it's more important than trying to pre-plan carefully, cerebrally, the motion of each of its legs. As you can see, it leads to a much more stable implementation of movement. And it's not pretty yet, not like a gazelle that runs across your backyard, but most probably in a few more years these robots will be quite graceful, because probably once they optimize the motion, they will seem graceful, because when something is done well, it usually looks pretty to us. But now the question, remember those circuits that generate a repetitive motion and can activate different muscles in a patterned way so that we could run and walk? How do they work? I won't be asking this question in a ping mode because it's kind of hard. You probably get the point that in order to generate a loop, you probably need to have something connected in a loop. So you need some process, trigger some other process, trigger some other process, trigger some other process. And there are different solutions for that. You can make it so that everything is within one cell. There are cells that generate patterns on its own. For example, you can have a cell that generates an action potential, gets inactivated, goes down, goes through a refractory period, depolarizes, and again generates an action potential. You can have a neuron that just spikes every few milliseconds repetitively without any help from the outside. You just need to make it depolarize after the refractory period. Just introduce basically a sodium current that's always on, and you have it. One example of a cell that works like that, which is not actually a neuron, but a modified muscle cell, is the cell in our heart. And cells like that are called pacemakers. Pacemaker is a key word here. Pacemakers are cells that generate this repetitive activity. And remember when we talked about experiments where acetylcholine was discovered, if you cut the heart out of the body, it continues beating. It turns out that if you cut these cells from the heart, they continue producing the rhythm even in a petri dish. They generate the rhythm and then it spreads all over the heart and makes the heart beat. Another example that's similar but works at a very different time scale are circadian rhythms, those daily rhythms that we have. Not even sleep-wake cycle, but how we're more active and feel more alert during the day. And all the things that change, the blood pressure or the temperature, the hormonal status, all of that cycles every day. And there are cells in our brain that control that, which is a very interesting story that we won't be discussing right now. Or you can have a bunch of cells, say a bunch of neural cells activating each other. So maybe you have one cell, it activates another cell, it activates another cell, and so activation just goes through this network again and again. And conceivably, you can also have a third option. You can have a system that generates an output that changes the world around you to such an extent that it stimulates you again and you again generate a certain output. It's hard to understand if I put it that way, but for example, flying in bees is actually not like running in humans. In humans, you have a bunch of cells that generate repetitive activation of muscles. In case of bees, it's slightly different. You have cells that stimulate wings. Wings move, it activates some other cells that again stimulate wings. It's actually not wings, it's a little bit fancier. They have a special spring to which the wings are connected, so the muscles don't move wings directly as in dragonflies. But still, the idea is that you move something, it affects your body, gets detected, and leads to a new wave of activation. Another name for that is chain reflex. When one reflex causes something in the world or in the body, that immediately triggers another reflex. But let's concentrate on this middle solution because this is the most famous one and the most important one. And this situation, when you have a bunch of cells that together as an isolated circuit can generate a repetitive pattern that leads to emotion, is called a central pattern generator. Let's talk a little bit about that. Central pattern generators were discovered by Yves Marder, who is one of my scientific heroes personally, a very famous neuroscientist. Central pattern generators were first discovered in a stomatogastric ganglion. And it is a funny object it's worth talking about. In crabs and crayfish and lobsters and all of these decapod crustaceans, there is a thing called a stomatogastric ganglion. When a lobster eats, when a crayfish eats, it chews on food, kind of like humans do. But unlike humans, it doesn't chew on food before swallowing. It first swallows food, then it chews on it, because it has teeth in its stomach. And that's fun, isn't it? It has these teeth, that's how they look like, inside the stomach. It first catches the prey, it swallows it, and then it chews on it and grinds it to a nice puree that can be digested later. The movement of these teeth is controlled by a tiny ganglion that is located near the stomach, called the stomatogastric ganglion. It's a bunch of cells. From these cells, the fibers go to all sorts of muscles that control the movement of the stomach and the movement of these internal teeth. What people realized is that you can cut it out. It continues generating these rhythmic patterns. But because it's a crab or a lobster or a crayfish, you can cut it out and it will live in the petri dish for days. The preparation itself takes a few hours. However, if you manage to perform it correctly, you can experiment for a really lot of time. It's cold-blooded and it's an invertebrate, so from the ethical point of view, it's awesome, right? It's very well studied by now. What cells is this ganglion made of? You can see some of the cells on this picture even, right? Each of the cells can be named. That's one interesting thing about many invertebrates and many systems inside invertebrates. In us, in humans, if you look at a certain part of the brain and you will find some cells, there is no guarantee that another human will have exactly same cells in the exactly same part of the brain. We are much more generally built, so to say. We have like cells of this generic type in this generic area. That's the level of replicability from one human to another. In case of stomatogastric ganglion, if in this species it has 32 cells, then in any other individual of this species it will have 32 cells. And if the cells on the left works with serotonin, then in any other individual of this species, the cell of the left would be releasing serotonin, which again makes it a really attractive model for studies. By now we have a basic understanding of how they're connected, and all the cells have names now. See, pyloric neuron, gastric meal neuron, gastropyloric neuron. There is a bunch of electrical synapses and chemical synapses. There is glutamate, there is acetylcholine. I think there are some other neurotransmitters as well. There are lots of neuropeptides that modulate its function. And if you record from them, even from an isolated ganglion, you have these periodical signals. Different neurons are doing different things. Some spike later, some spike earlier. And the key pattern, the key mechanisms on which it is built is recurrent inhibition. Why recurrent inhibition? Why not recurrent excitation? When I first read about that, I thought, okay, recurrent excitation should be the thing, right? So one neuron activates another neuron, it activates another neuron, and it activates another neuron. If you have a cycle like that, activation would run in a loop. The problem, though, is that everything is really fast. Action potentials first appeared in order to facilitate communication between different parts of the body. The whole idea of an action potential is that they're fast. If an action potential takes a millisecond to generate, it's a millisecond here, it's a millisecond here, it's about 2, maybe 5 milliseconds here. You can generate really fast rhythms with that. If you have two neurons and they activate each other, every 10 milliseconds this neuron will be activated, assuming 4 milliseconds for the synaptic transmission. Every 10 milliseconds is 100 times a second. It's very fast. In some cases, you may want a circuit like that. Maybe if you're a fly and you want to move a wing. But if you're a human and you're running, you want to have how many? 2 cycles per second? How often do we move our legs? Something like that, right? Maybe I'm off a little bit, but not too much. Definitely not 100 times a second. You could, of course, just have more and more neurons in this loop, but there have to be better ways, right? We want to slow it down somehow. I guess one could think of maybe metabotropic excitation, but for some reason that's not what's happening in nature. Maybe there is some complication here that I don't see immediately. But what happens in nature usually is recurrent inhibition. So you have two neurons, at least two, that inhibit each other. These inhibitory synapses. When this neuron spikes, this neuron doesn't. But then at some point, this neuron gets tired. Tired goes in quotation marks, but we somehow built in that it spikes and then in a while it stops. When it stops spiking, this neuron is no longer inhibited. And now the only thing that you need to add is that this neuron would start spiking on its own. So it was inhibited, now it's not. It wants to start spiking on its own. It's well rested. All of that goes in quotation marks. It depolarizes until it starts spiking, and now this one is spiking, and this one is forced not to spike. And they will change these roles again and again and again. To just create a rhythm, it's enough to have two neurons that inhibit each other. If you want to move one muscle, and then another muscle, and then another muscle, you want to generate several phases of the same rhythm. So two neurons is probably not enough. 2 neurons is barely enough to have two muscles that are activated one after another. But if you have more, then you probably have more neurons. If you look at these responses from a stomatogastric ganglion on the left, you can see that these two neurons are active together, but this neuron is active before that, and this neuron kind of after this one, but before this one. So depending on which of these neurons on this diagram you connect to which muscle, you create a pattern. This muscle is activated, then this muscle is activated, then this muscle is activated, then maybe we miss a beat and we go again. 1, 2, 3, 1, 2, 3, 1, 2, 3. you can create all sorts of patterns that are hardwired into the system. And you can move your legs and wings and tails and whatever you need to move. Stomatogastric ganglion, internal teeth. Lots of things that we notice in a spinal cord also happen in the stomatogastric ganglion. And in fact, they were first discovered in the stomatogastric ganglion and then found in the spinal cord. This switching between walking and galloping. In this internal stomach of a lobster, same thing happens because sometimes it chews and sometimes it mixes food and sometimes it swallows. It's like 3 different modes of operation. And when we walk and run, we also switch these circuits from one mode of operation to another. It's not actually 2 different circuits, it's one circuit that can just be switched from one mode to another, from one gear to another using modulatory neurotransmitters. In a turtle, for example, the same circuits that can move its legs for crawling can be used for swimming, or they can be used for scratching its body. And to conclude this lecture, think of this question. Guess what other processes and behaviors could be driven by CPGs. So we talked about walking and swimming and running, and also about this internal grinding of food inside the lobster. What other behaviors, what other motions can also be guided by central pattern generators? And remember that we don't include in this list heartbeat in a human. It's similar but different. It has a pacemaker. And we don't include into this list sleep-wake cycles and circadian cycles. They're also controlled by a central pattern generator in our brain. What else can you think about? Typically, people can come up with like 5 or 10 examples, so try to figure it out. Pause the video, answer this question, then compare it to my list. Don't cheat, pause the video. All right, let's compare locomotion of all kinds. We talked about running and swimming, but of course slithering, flying would also count, right? Any kind of locomotion that includes periodical motion of something would probably be controlled by central pattern generation. Chewing. This thing that happens inside the lobster is essentially chewing. It's just not in the mouth, but we chew in the mouth. That's okay. It's also acceptable. Drinking. If you imagine coming back home from a walk on a really hot, really dry day, and then you get to the cold water and then you drink and you're like gulp, 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 gulp. This motion is controlled by central pattern generator, obviously. Or at least it feels like that. I think it is. Scratching. That's interesting, you know? Like, if you start scratching, you can do it unconsciously, and especially when you do it unconsciously, then you immediately realize that it is a periodical motion that is almost like controlled by someone else because it's in the spinal cord. It's controlled by your brain, but it can also very happy to delegate it to the spinal cord if it can. Grooming. Grooming is kind of similar to scratching, but it's more like cleaning yourself or doing something like that. Usually grooming is used when we talk about mice or dogs, or if you've seen flies cleaning their back legs, like trying to clean themselves. All of these motions, all of these behaviors that are related to cleaning the body of dirt and parasites is called grooming. Copulation, well, you might have got this one. Respiration, which is breathing. Breathing is an obvious one, right? And that's a good example of repetitive behavior that we don't control consciously typically, but we can. Humans can. Many animals, most animals actually cannot control it consciously. They cannot stop breathing for a second. Some animals, like dolphins, cannot not do it consciously. They don't have a CPG that controls their breathing. They only do it consciously, which makes sense if you think of what can happen if dolphin suddenly starts just breathing on its own underwater. So dolphins should not lose consciousness ever. If a dolphin or a whale loses consciousness, they may die of suffocation because the only way for them to breathe is to think about it and to do it consciously. There is also a condition in a human called Undine curse, when humans lose the ability of breathing without thinking about it and can only breathe voluntarily. And then it's hard for them to sleep, obviously, because they would die without a ventilator. So they have to sleep on a ventilator and they have to remember to breathe all the time. And it's called Undine curse, after a medieval story about a mermaid who cursed her unfaithful lover to death by removing from him an ability to breathe involuntarily. Coughing, probably, yeah. Sucking in babies, also periodic motion. Heartbeat, definitely not in a human. You wouldn't call it a CPG because it's a pacemaker. However, in some animals, in a crab, for example, the heart is controlled by a bunch of neurons and they form a CPG. Circadian rhythms, probably not, but similar. Sniffing, so sniffing is this thing that some animals do, and that's totally controlled by CPG. Whisking. Whisking is something that animals with whiskers do, and what they do is they do this motion when they investigate an object. They don't just sniff it, they also touch it with whiskers, and it's a periodical process controlled by CPG. Sound production. Not in us. When we produce sound, we use a different system, but then there are animals that produce sound through a periodic motion, like purring in a cat, for example, or all sorts of chirping, or like sound that some birds like a heron do, croaking and a frog. So if there is a periodic activation of a muscle, it has to be CPG. Courtship and flies, remember? Extend the wing and move it. Sensing in electric fish, that's fancy. You can read about it. Electric fish use modulated electric field to investigate what's around them. Struggling. Struggling is an interesting behavior. It's when you catch a fish and fish does this special motion to escape from you and get back into the water. It's called struggling. It's an official name for this. It's also controlled by CPGs that kind of switch from swimming. It's kind of like slithering, but extreme slithering from a predator who is trying to catch you. Shaking, when a dog gets out of water. Not shaking that humans do when they're cold. That's actually different. But shaking water off in a dog. Any activity of the digestive tract in us, this peristalsis, moving the food along the intestine, it's probably also CPG. I mean, it's definitely CPG. It's just of a slightly different kind because it's a different part of the nervous system. It's not the brain, it's not the spinal cord. Gut has its own nervous system called enteric nervous system, special bunch of neurons that only controls the movement of food through the gut and its processing in the gut. It's surely an incomplete list, but it gives you the spirit. And if you have some other good ideas, please contribute in the comments maybe. This concludes our lecture. Thank you. See you later.
Let us learn about the brain.